But today I concentrate mainly in the neutrophils. I have to make a disclaimer that I'm not an immunologist, but I have very nice immunology around me, so that's why we are doing this kind of investigations. So I don't think it's necessary to explore deeply this slide to this audience, but one thing that I would like to call your attention it exactly to this, what happens here in between this partially or misfolded state and the prefibrillar and the amyloidal mature fibrils. So what the species that accumulate here that we already know, and Thiago just mentioned us how these species that are present in this specific step of the aggregation process, how they are toxic and how they have different shapes and architecture. And so they can exert several bad functions. And Thiago again mentioned that it can impairs long-term potentiation. It's just one example. And I would like to call your attention to the fact that the amyloid fibers already done, already made, mature, they can undergo what we call amyloidolysis. That's the process of the breakage of this structure, feeding and again this toxic species. And so in my lab we got interested to understand more deeply what would be in vivo, what could faster this process. And in this case, we decided to investigate this specific cell from the immune system, that's the neutrophils. So how, and I'm going to explain you what the data we got in our lab, showing how these cells can break and can feed these species in vivo. And of course, these species, they are very important because they can spread and they can act like a prion-like species. So again, you don't need any more explanations how important this fibril structure, how they are in all these diseases, the localized or the neurodegenerative diseases. And and I got interested into study and to make any correlation in relation to neutrophils and amyloid fibers. There are very, very few and rare papers published in the literature making this connection, but I got very interested specifically in this kind of old papers from the 90s. From the Martha Skinner's lab, showing that in different types of amyloid diseases, amyloidosis, the primary, the TTR related, the beta-2, and all these amyloidoses, when you look close to the deposits, you can find proteins from the neutrophils, namely elastase and cathepsin G. So showing us that probably in these deposits, they can call the neutrophils to the site of the deposition. So, and then I got a little bit interested to understand deeply this correlation. And then I start to work with these very interesting cells that are the neutrophils. So the neutrophils, they are very important because they are like the infantry of our innate immune defense. So when we have any problem, an infection, so the neutrophils are the first one to be recruited to the site of these infections. And neutrophils, they have several ways to help us and to get rid of the invaders. Just just as phagocytosing the microbes or the granulating, and all this enzyme content to the sterol in order to kill the microbes. But recently, the Ziklinski group in Germany, close to Thiago's lab, so they described a new, very interesting type of cell death that they call netosis in relation to apoptosis and necrosis. That's the way the neutrophils, they die, and during this dying process, they release what they call the neutrophil extracellular traps that I'm going to call since now NET. So these NET are mainly composed of DNA and decorated with all the enzymes, the weapons that these cells, they have to kill the microbes. And so it really works as a net when you fish and you trap, and you kill the microbes, and when they are trapped in these cables, in this web, so like the Spider-Man. So since this description of this net, so there are several works trying to understand better this process in the physiological relevance of net formation in the real world. And there are several studies showing beautiful and interesting picture of the neutrophil expelling these webs, and you can see all the microbes trapped, and they are really killed inside these nets. So just briefly, because we already know a lot of things about net formation, but still there are several gaps to understand all the process related to this new death mechanism. So we do know that there are several activators of the neutrophils, like microbes and IL-8 and LPS and activated platelets. So all these are net triggers. And then we do know that the ROS production by this enzyme, the NOX2, that's assembles at the plasma membrane of the granule membrane, they produce ROS species that are very important, at least to a special type of net formation. Then in the third step of this process, and elastase and myeloperoxidase, they migrate to the nucleus, where there undergoes a process that's chromatin decondensation added by this enzyme. That's the amynase, the peptidyl arginine, the amynase that cancel the positive charge of arginine, so then releasing the chromatin structure, and then we have the nuclear membrane disruption, and then finally you have this interesting extravasation of all the content of the cells in the shape of a cable. So when we start to work with this system, so there are many studies showing all these microbes that are net inducer, and net was also correlated with several sterile inflammations such as sepsis, cancer, and so on. And our first approach was to pose us a very simple question. So is the amyloid fibril a net inducer? So remembering that Martin Skinner's group reported the presence of neutrophils enzyme very close to the site of amyloid deposition in these patients. So then these very talented students from my lab, Stefania, she now is in Rockefeller University. And so we produced in our lab several different types of fibrils, from transthyretin, synuclein, sub-35, that is the prion protein from yeast. And the idea was to have different amyloid structure with different primary sequence, and also we produce, just changing the conditions, the amorphous aggregates from the same proteins, and then we look whether we could see extracellular DNA that was our readout of net formation. So, and then here I'm showing you the images of what we got. So as you can see, so incubating these neutrophils with all these fibrils, we could see the presence of these nets. I don't know if you can see, but they are positive for elastase. DNA, we can see these clouds that resembles what is described in the field. And one thing that called our attention is that the amyloid fibers, they induce net formation, but they also get close to and trapped and adhere to this DNA cable. And this was very specific to the fibrillar form of this protein, because when we add amorphous aggregates, we can see that the DNA is confined inside the neutrophils. This lobular shape is very typical from the neutrophils DNA. So here's just to show you a very nice transmission, high resolution transmission electron micrography that with unpublished data from our lab. So this is beta fibrils. So you can see here the neutrophil completely destroyed. And so the cables that I just mentioned for you, that's the net. And then you can find this small structure here that we think are the A beta fibrils. So again, these fibrils are composed of the A beta is also able to release net. So our next question was to see, okay, nets released by the amyloid fibers, but what is the interplay between, so since between the nets from all the components that are present in nets and proteinaceous material as the amyloid fibers. So what we did to tackle this question, so we incubated the neutrophils. So I forgot to mention that PMA, the 4-ball metastatic acetate, is the classical net inducer. So you incubate the neutrophils with this pharmacological agent, and then you purified only the net component, and we incubated this net component with the fibrils, and we went to see what happened, since there are several proteases associated with this material. So we did several experiments. I'm just summarizing some of them here. I just want you to see the images. So before digestions, we have the typical mature amyloid fibrils here. And then when we add what we call this span, the supernatant of PMA activated neutrophils, you can see completely digestion of this material. And this material is positive, as you see here. It binds the anti-oligomer antibodies, confirming or suggesting that these fibrils are being dissolved or digested, forming oligomers. And when we add the elastase inhibitor, we blocked completely this digestion. And here is only the elastase, the purified enzyme, also digests these amyloid fibers. And we think that elastase is one of the major component of NET that is attacking and digesting our fibers. So we took this material, these species that are present after the span digestion, and when we add it into a cell culture, as you can see here, it's killed completely all the cells, so no more live cells in culture. So showing that these digested material, they are very toxic to the cells. So since we are biochemists, so this is a new study that we did in our lab. So we investigated, so since this NOx enzyme depends on the production of NADPH, so this is the pentose phosphate shuntle pathway. So what we showing in this work is that in the presence of glucose, we do see formation of amyloid fibers when we add like synuclein fibers or PMA. But when we incubate these neutrophils in the presence of fructose that enters here in the glycolytic pathway, so you cannot feed anymore the pentose phosphate pathway, so we cannot produce net formation, showing again the importance Okay, Thiago, again, the glucose is very important to, in this case, again, to the amyloid fiber induced net formation. So, and I'm going to skip this. And so, and then on the next step is to see whether if there were any physiological relevance for all these observations. And then we went to see whether we could, we found slices of patients, post-mortem slices from patients that died with primary amyloidosis, lung and skin. And as you can see here, we could find NET, the presence of NET in these slices. So they are positive. You can see here, I don't know, but so you can see this cable, these clouds of DNA that are positive for elastase, for histones. And so they are positive for thioflavin S, confirming that everything is there in the tissues together. And here we can see also the formation close by of this deposit. I don't know if you can see, but it's very faint. I do agree that the presence of the oligomers in these tissue sections, confirming that everything that we showed before in vitro might happen in vivo and might have an implication for the courses of the amyloid diseases. So, and then we got very happy because just after we published our first studies showing the implication of net formation in amyloidolysis, that's the process of the fibril breakage. So this paper, this very wonderful paper, was published by the group of Gabriella Constantini from Italy, showing the presence of neutrophils in the parenchyma of the Alzheimer's transgenics animal models. And as you can see, this is pink dots. And interesting, when this group blocked the entrance of the neutrophils with this antibody against these integrins that's present in the neutrophils cell wall, so they could ameliorate the cognitive decline that these animals have just by being transgenic. So if you impair the entrance of the neutrophils into the brain, so you can alleviate this cognitive decline. And then when they look this in tissues, brain of vessels of AD patients, again, they could see the presence of NET in this patient. So confirming that indeed this process is important in this context. So I want just to go a little bit further. So these are more unpublished and new data that was just collecting before the pandemic. So we have to stop this project, but I decided to bring it to discuss with you anyhow. So one thing that I want to point out is that everything that I mentioned, almost everything, is that showing you how NET could be, there is this good side of the NET formation. It traps microbes, it kills the microbes, but I just mentioned you that it digests amyloid fibrils into toxic species. So this is the other side of this double-edged sword. And there are several reports in the literature showing how NET could damage the peripheral tissues and cause autoimmune disease, because if it persists longer than it's necessary, all these enzymes start to attack the peripheral tissues around where the site where NET is formed. Also, we have to remind that in this NET webs, so we are exposing our DNA. So it's a material that can elicit autoimmunity, and indeed, this is being seen in lupus erythematosus, that's an autoimmune disease. It's completely associated with this NET formation process. So we have an enzyme that's called DNase that circulates in our blood, that has its function is to digest and dissolve and resolve this NET structure in our body. So just show you something interesting data, again, not published. So we have this cohort of FAP, familial amyloid polyneuropathy patients. And as you see here, so they have several stages of the disease, FAP 0, 1, 2, and 3, and we evaluate the DNA's content in the plasma of these patients. And as you can see here, so the health subjects, so they can digest completely the net when we add this plasma into net in vitro, but some of these patients, they cannot digest. We have around 60% of non-net, non-degraders. So, and we envision that in this case, these patients that has deficiency in resolved net, so they have a persistence in this structure that keep digesting the amyloid fibrils and keep releasing this toxic component. So this is ongoing studies that we have to confirm whether these DNAs in these patients, they don't have the level of the DNAs in their plasma is decreased, or whether the protein is there, but there are some activity inefficiency. So we are studying this right now. So I guess I have just more 5 minutes. I'm going to close very soon. So, and then that's again unpublished data that I want to share. So since we noticed the presence of amyloid nets formation in the lungs of a primary amyloidosis patient, so we decided to make a homemade amyloid animal model in our lab, and we were very fortunate because we have this knockout animal that has a deficiency in this subunit of the NOX, and in this animal they cannot produce net. So we have on our hands an animal model that we can prove the importance of net during the course of amyloid disease and amyloid formation. So as you see here, these knockout animals, when we add PMA, they do not release ROS. So as expected, the wild type animal, they do release, they are neutrophils in vitro, they respond to PMA, that's a net inducer, and increase the production of ROS. And here, as you see, when we add alpha-synuclein fibrils to the neutrophil from the wild type animals, we could see this again, these webs, DNA labeled webs. The knockout animals, they cannot form net unless we add H2O2, oxygen peroxide, and then we compensate this the loss of this enzyme, and then these animals, they can release nets. That's the only problem they have. So, and then what we did, we instillated amyloid fibrils into the lungs of this animal. And then first we saw whether this knockout animals, the wild type and the knockout GP91 Fox animals, they could recruit neutrophils, and they both, when injected with fibrils of alpha-synuclein, they can recruit, no problems with the knockout animals. But as you see here, these animals, they, of course, when we inject fibrils, their lungs are very inflamed because the presence of the fibrils, but a lot of collagen production. But one thing that we noticed, that the presence of this structure here much more abundant in the knockout animal, that was almost completely absent in the wild type mice. Just to expand a little bit the image, you see these sticks here, yellow, when we stain for collagen, that were only present in the wild type, in the knockout mice. And so we thought probably these animals, since they cannot release net, they are accumulating these fibrils. So, but we didn't know whether it was really amyloid fibers that we instillated into the animal. So we took advantage of the Congo red. And again, as you see here, the GP fox animals, they do bind Congo red, but not in the wild type lungs. So confirming or reinforcing our idea that these instilled fibrils, they disappeared somehow in the wild type animal, but they keep there in the GP fox animal. So then we probably is the net. Let's see whether these animals, they are forming nets. And so here, just to show you, slices of the lung of these animals, is the wild type, injected with the fibrils of a synuclein, but we can inject any fibrils, so any model can elicit this process that I'm describing to you now. Here, the model was synuclein, but we can inject other fibrils that they act more or less the same regardless the primary structure. So we do see the presence of histone citrullinate, that's 2 myeloperoxidase. These are 2 net markers, but in the knockout mice we couldn't see the formation of net, and suggesting that again that this probably this the net that are being formed in the wild type animals, they are being responsible for the cleaning of the digestion or elimination of these fibers. And just to show here, this is a physiological pulmonary function, so that we are evaluating right now. So we have only this data with the wild type mice. So here we use methacholine, that's a bronchoconstrictor, and so we challenge this animal with this compound. So here is only for the wild type mice. I don't have right now the data, the respective data for the knockout mice, but just to show you that in the presence, so in the saline, that's in white, no problem, the animal stands the methacholine injection, either measured by elastance or resistance, but in the presence of the fibrils, so 3, 6, and 8 hours, they do have, in the presence of higher concentration of this bronchoconstrictor, they do have problems. But as the fibrils, they probably are being digested, so they recover this functionality, this lung functionality, and are very eager to see what's going to happen, whether this is going to be kept elevated in the wild type mice. So just quickly, I'm going to skip this, but just to show you how NET is could be very toxic. So this here is a model for the FAP. So this is a dorsal root ganglia, that's the peripheral nervous system ganglia. And so here's the control, and I want just to call your attention. So here we add increasing amounts of nets. We can see that all the neurites, they grow in culture 3 days or 5 days. They are a little bit damaged due to the time these ganglia are in culture. But look how net is very toxic to this ganglia, to this neuronal cell. So here I'm showing you another model. So I just showed you the lungs. Now I changed to, and so the neurite lens is completely disturbed by the presence of net. The area that they occupied, they are very toxic. So presence of nets are very toxic to these cells. They died completely. But in this case, we were very fortunate because we use DNAs to disrupt net, elastase inhibitor, and myeloperoxidase inhibitor. And as you see here, DNAs, the addition of DNAs that digest net didn't change the profile. So in the presence of net, the ganglia are very damaged. In the presence of DNA elastase inhibitor, again, it's a little bit better, but still very compromised. But in the presence of myeloperoxidase inhibitor, we could recover almost completely the neurite lens, and we could abolish almost completely the toxicity of the net. So in this case, we could target, we can say that myeloperoxidase is the net component that's very toxic to these neuron cells, what opens possibilities for intervention in the case of FAP, where we find amyloid fibrils just in the peripheral nerves. This is my conclusion. So with all that I showed you, so what we think, so we have an amyloid deposit so we have airways commitment or peripheral nerve commitment. So these fibers, they recruit the neutrophils and other inflammatory cells that we evaluated, but I didn't show. I showed you here. So it causes a severe inflammation in the tissue. So, and these fibers induce net formation, and this net formation, all these enzymes present in the net, they digest and fragment the amyloid fibers. And so in the lung, once these fibers are digested, so we can see lung restored, the functions of the lung are restored. But in the case of the peripheral nerve system, we can see that these fragments, they are, we can see that these net components, they are very cytotoxic, and we are still evaluating what happens in the lung in the presence of this net component in relation to cytotoxicity to the tissue around. And just finishing, so this is a recent paper from a Brazilian group, so showing the COVID-19. So in COVID-19, SARS-CoV-2 induces net formation in the lung, and it's present also in autopsies of the patient, so showing the importance of this net structure in so many diseases. So I just want to close. Thank you again for the opportunity, and these are the collaborators, the students so all my students in my lab, but specifically these 3 students that work directly in this project, and the state funding agency, mainly FAPERJ, because the other 2 are not giving us money anymore in Brazil. So thank you very much for your kind attention, and sorry for the 2 minutes that I